This tutorial will focus on chromosome mutations. There are two kinds here. One is you could end up with an extra chromosome to the normal, or less than one chromosome compared to the normal, or you could end up with broken chromosomes. Maybe part of a chromosome is broken off and rejoined to another one, or a chromosome may have had a bit added to it. So we'll have a look at these different scenarios for chromosome mutations. Well, first of all, start with just a few terms that are important to understand. First one is aneuploidy. An aneuploidy refers to simply having an abnormal number of chromosomes. So an abnormal number of chromosomes appear in your cells. Abnormal number of chromosomes. The most common condition of this is trisomy, where you end up with 3 copies rather than 2 of a particular chromosome number. Aneuploidy usually occurs at some stage during cell division and generally involves a process called nondisjunction. And nondisjunction simply means that a homologous pair of chromosomes does not separate during the first stages of meiosis. So in anaphase 1, a pair of chromosomes doesn't separate. So let's have a look at this process on the right, and it shows this process happening. So on the right we have normal meiosis in a male. So it doesn't really matter what the pair of chromosomes is we're interested in at the moment. Let's say we have 44 chromosomes plus another 2, so this is our 46 chromosomes in a germline cell about to undergo meiosis. Normally what would happen is these chromosomes would divide into half during meiosis 1, so you'd end up with 23 chromosomes, in this case 22 plus the one we're interested in each of the cells, the 2 daughter cells at the end of the first division of meiosis. So by the end of the first division we have 23 chromosomes in each of the daughter cells, and then they just go through the second division to end up with 23 chromosomes in each of the sperm cells. So that would be the normal situation. Now on the left we have abnormal meiosis in a female. So same starting point, 44 plus 2, so this is our germline cell here. But during meiosis 1, this pair of chromosomes here should separate, so each member of the homologous pair should end up in a different daughter cell. But in this case, they end up in the same cells. So this pair of chromosomes here does not separate during anaphase 1 and both end up in one daughter cell. So you end up with 22 plus 2. So you end up with 24 chromosomes here, and this daughter cell is actually missing a chromosome now, so it only has 22 chromosomes. Then they go through normal meiosis 2, and the egg cells that are produced are going to have an abnormal number of chromosomes. They're going to have 24, 22 plus 2, 22 plus 2, and from this daughter cell here, are only going to end up with 22. 3 of the ova become inactive, they become polar bodies and degenerate. That's normal. So these 3 would degenerate. This one here has 24 chromosomes. If this egg cell here then fertilizes a normal sperm cell, in normal fertilization, this process here, they produce a zygote that has an abnormal number of chromosomes. Remember, the normal human cell would have 46 chromosomes. That would be 23 pairs. In this case, there are 47 chromosomes, 24 plus 23, so 47 chromosomes. So would there be 22 pairs, plus there would be 3 copies of one of the red chromosome in this case. So you'd have 2 from the mother, 1 from the father. So this is a case of trisomy, that is 3 copies of a particular chromosome. This, for whatever reason, seems to most commonly happen with chromosome numbers 21, 18 and 13. If you end up with trisomy 21, that is 3 copies of chromosome number 21, then you have Down syndrome. Trisomy 18 is known as Edwards syndrome, and trisomy 13 is known as Patau syndrome. Downs was probably the the most common one that people are familiar with. So just a quick reminder, nondisjunction means the non-separation of a homologous pair of chromosomes at anaphase 1. So the chromosomes do not separate. Another term you had to look at was monosomy. And monosomy simply refers to the lack of one chromosome of the normal complement. So you may only end up with one chromosome rather than two in your normal diploid complement. Let's have a look at a couple of other kinds of chromosomal mutations. Let's first of all look at a duplication mutation. To duplicate something means to make an exact copy of it. So a duplication mutation sees a portion or segment of genetic material, say this bit shown in this diagram here. So it shows a portion of genetic material which is duplicated or replicated, and it results in multiple copies of that region. So you end up with a second copy of it here. So when the chromosome replicates, you end up with two copies of a particular region. So you end up with a duplication mutation. It is possible in some circumstances that an entire chromosome could be duplicated, so you end up with an extra copy of the chromosome, but perhaps more often than not it's just a portion of the DNA that gets duplicated. So let's have a look at the rest of the chromosome mutations, or block mutations as they're sometimes referred to. So this involves parts of chromosomes usually. So we've just talked about the duplication mutation. In this particular diagram, we can see here that the BC segment has been duplicated so that it appears twice after the process. There's also a deletion mutation where a segment of, now this is not the deletion of a single base, this is the deletion of a segment of a chromosome. So in this case segment D is deleted from the chromosome, so genetic material is actually missing from the resultant chromosome. Here genetic material is repeated, so you have two copies of it. There's an inversion mutation where a particular sequence of a chromosome, let's say BCD is shown here, can be inverted, turned back to front. You end up with DCB. You end up with the same genetic material, but it does depend a bit on whether genes are disrupted in this process. And you've got translocation. Now translocation is a breaking off of one bit of a chromosome and joining to a different chromosome, generally from a non-homologous pair. You can take a moment to pause the tutorial here and have a good look at translocation, but we'll have another look at another diagram also in a minute. So here's another diagram of those, deletion, duplication and inversion, but here it shows translocation perhaps a little more clearly. Probably the textbook has a good drawing on this as well. But here you can see two chromosomes, number 4 and number 20, so non-homologous chromosomes, and a bit of chromosome 4 has broken off and joined to a part of chromosome 20, shown here, and a bit of chromosome 20 is broken off and joined to chromosome 4. So there has been a translocation of genetic material, and so yes, you end up with bits of different chromosomes joined to others. The consequences of different kinds of translocations depend on what's actually happened and what ends up in the gametes. In normal or balanced translocations, so where basically despite the bits that are broken off and rejoined to different chromosomes, as long as all the genetic material ends up present in a particular gamete, then there's no loss of functionality. In other words, all the genes are present and they can be expressed normally. Where there's an unbalance in the gametes, so depending on the way that the chromosomes have segregated during meiosis, it's possible you may end up with gametes that are missing some genetic material or having extra genetic material, and that generally has consequences for the expression of the genes and the consequences for the creature, the organism, which is not always good.